All right, ladies and gentlemen, here we go with learning and memory. We're off to a brand new topic again. There are going to be four subsections that I'm going to break into two lectures for you. A lot of people like the learning and memory lecture, but there are some tricky nuances to it. So if you need help, we're always here to help. So as I said, the first thing that we're going to talk about is memory. There are a few different stages of memory that we need to talk about. So the first thing that happens for all of us when we're trying to process information is that we form a sensory memory. And this is a very, very short memory that lasts at the most just a couple of seconds. So this is the sort of memory where if you picture something and you close your eyes, you can retain a picture for a few seconds. And the way that we think that this works is that there is a little bit of a sensory echo that happens when your eyes or ears or something else has processed this information. You're able to sort of play that back for a few seconds. For things that we need to remember, though, we often put them then into short-term memory, and this is information that we can remember for a few minutes, usually, maybe a little bit longer than that, and there's a lot of individual differences in the timing of short-term memory. You can remember this for longer if you do something like rehearsing. So for those of you that have been using cell phones for most of your lives, you're probably not used to doing this, but once upon a time, old farts like me had to remember everyone's phone number and walk over to write it down, or remember a phone number before we got to the touch tone telephone that we were trying to dial from. And so you would sit there and say it over and over and over again. Okay, I need to remember this phone number, 867-5309, 867-5309. That'll help you remember it. That's a form of rehearsing. And you can try to remember things using that. And for short-term memory, we can also only hold a limited amount of information. So that's one difference between what we think of as short-term and long-term memory. Long-term memory are the memories that last theoretically for a lifetime. And this really depends on the memory, and we need it to be consolidated. So we take something that was a short-term memory and we actually have certain things that happen within the brain in order to take that short-term memory and turn it into something that we've permanently stored. And when you all say, oh, I remember this, or I have a memory of when, we're almost always talking about long. One thing that you need to know is that long-term memory also takes multiple forms. So there are declarative memories, and these are the things that you all probably think of when you're thinking of memory. Declarative memories are the stories that you can tell about a time that you went to a certain place, or even the facts that you remember from, exam one of physio psych, or your first semester at Rutgers, the class that you really loved. All of these fall into a category of declarative memories. But there's also a set of non-declarative memories. And so these are things that you have developed some sort of a skill for, like riding a bike, or something else that you've stored long term, and you can show that you know these things, and you can show you've gotten better at them, and that's usually how we test for them. But they don't fall into the same category where you can necessarily tell somebody using some sort of a story or an anecdote what it means to recall that memory. You usually have to demonstrate them. So all these different forms of memory bring us to learning about different forms of learning. And the reason for this is that different parts of the brain are responsible for executing or processing different types of memories. And this is where that long-term memory breaks down into multiple different subgroups. So one of the earliest experiments, in one that we might have even talked about during our introductory lecture where we talked about the history of psychology, were experiments in mazes that were done by a psychologist whose last name is Lashley, and he has mazes named after him. And part of the reason that Lashley is is so famous is he spent a long time doing brain lesions, or he would even take out pieces of the brain and remove even large pieces of of the cortex of mice and rats. And he was trying to find the seat of memory. And all that he ever found, which is in some ways an amazing discovery in and of itself, is that the more brain he removed, the worse memory would get. But he could never find the seat of memory. And part of this is because he would have had to have taken out multiple systems in order to fully eliminate memory, and especially to eliminate the many different strategies for memory. Stimulus response learning takes two forms. And if you've had Intro to Psych, you've probably heard about both of these. But as I always say, my assumption coming in is that everybody knows nothing, and I'm going to start from the bottom. The first of these is classical conditioning, and this is where an involuntary response, or sometimes even people would call this a reflex, is paired with some sort of a stimulus so that you can get the stimulus to eventually evoke this unconditioned response. And operant conditioning involves mice or rats or even people it involves the need for them to make a voluntary behavior, which then leads to some sort of a consequence. And so this is a learning of action and consequence. So one thing that I want to do to get us all on the same page is to just give you a textbook definition that hopefully will help you to understand the difference between classical and operant conditioning. And you know this is not typically my style to read off definitions, so hopefully you'll bear with me this one time. But classical conditioning is when we have an association between two stimuli, so that a previously neutral stimulus, meaning that it's something that was meaningless before this, is conditionally associated. So this means that one thing is a condition of the other. This means there's a relationship between these two things, or some sort of a contingency where one must predict the other in time or space, for example. And so this neutral stimulus becomes conditionally associated with an unconditioned stimulus. So something, if you think about unconditional love, this happens without any conditions necessary, so that the neutral stimulus eventually becomes able to produce the response that would normally be produced by this unconditioned stimulus. So we start to pair things together to where they become able to produce a similar kind of response as the unconditioned stimulus used to do without needing any help. For operant conditioning, this is more a process where some sort of a behavior is going to be increased or decreased. And when we say increase or decrease, we mean in frequency. So you're going to come to class more or come to class less based off of the consequences. So if I were to give some sort of incentive, like giving out exam questions for people that come to class, then people might increase the frequency at which they come to class, for example. This is the sort of increase or decrease that depends on the behavior. As a comparison of these two, when we think of classical conditioning, as I said, a lot of people think of the behaviors as being some sort of reflexive behavior. The example of Pavlov's dog salivating for meat or meat powder, or whichever version of the story you remember or would like to believe. It's something that seems to be fairly automatic, these unconditioned responses, whereas in operant conditioning, they're most often learned behaviors. And in classical conditioning, we're talking about the association between two stimuli, this neutral stimulus and this unconditioned stimulus. Whereas in operant conditioning, we're really making an association between some sort of a stimulus, it can be a kind of an outcome, and a response. So because the response is automatic for classical conditioning, there's really only one outcome that can happen in classical conditioning. And this is whatever the outcome that was happening before was, whatever this reflexive behavior was. Whereas in operant conditioning, we can increase or decrease the probability that an animal makes a behavior depending on how we change the outcome. So it's a little bit more fluid. So there's a neurological basis for classical conditioning, and a lot of this might depend on exactly what sort of reflex that we're activating. A very common one that we understand really well is fear conditioning. So in fear conditioning, mice are put into a box and given some time to habituate and get rid of any stress or anxiety they have to being handled and put into a box. And then usually what happens is that there's a training day, or sometimes multiple training days, where a tone or some sort of a cue is paired in the box with shock. And so what would normally happen is that shock will produce some sort of a defensive response. And in a lot of cases, the idea that shock might be coming will cause animals to freeze, kind of like this deer in the headlights effect. And so what we've done here in a case of fear conditioning is that we've actually begun to pair the response such that now the tone, which is a previously meaningless stimulus, comes to predict this shock, right? This previously unconditioned stimulus, so that both of them can produce the same response on a test day. And usually that response is freezing. And what's been found over years of study is that, at least for this form of classical conditioning, it's very important that we have the function of the lateral amygdala. So many people, at least at a very basic level, think of the amygdala as a fear center, even though I've encouraged all of you to evolve your thinking a little bit and remember that no area of the brain is a fear center or a learning center or a happiness center. Many of these things process multiple types of information. But it turns out that in order to get this sort of association between these two things, at least at the level of a synapse, we need the lateral amygdala intact, which suggests that information from the somatosensory system relating to the freezing behaviors and the feeling of the shock is coming in, and this is being paired with information from the auditory system about the tone, and this all comes together in the lateral amygdala to eventually produce freezing. And it turns out if you lesion the lateral amygdala, then you won't get the freezing behavior at your test session when you just play the tone alone back as a way of trying to test whether this response exists. So what pairings like this have led to is a hypothesis that was first proposed by Donald Hebb. And this is a hypothesis that is still in many ways favored today, although there's some variations on the hypothesis. And what Donald Hebb proposed is that the way that we learn simply involves the strengthening of a synapse that is repeatedly active when a certain postsynaptic neuron fires. Okay, so this is kind of a longer definition, but a way that people often remember this is by this colloquial phrase, this simple kind of cliche phrase, where we say that cells that fire together wire together. And what we mean by this is exactly what Hebb said. If we have two parts of a circuit that are active at the same time, for instance, this input about the shock and the tone, and if those two are always activated at around the same time, then eventually those two sets of inputs to the amygdala will wire themselves together in a way to where they begin to cross-activate. In Hebb's rule, and the way he looked at it, would look something like this, where if you were trying to pair two things and you were trying to get them to wire together, what might normally happen, and an example that is used for Hebbian conditioning a lot, is an air puff to the eye. And so an air puff to the eye will normally make you blink. And in fact, there's a type of conditioning called eye blink conditioning that people use in all kinds of different animal models where they do this. They give an air puff to the eye and the animal blinks, and they do this over and over and over again. But this is a response that we all have. You know if somebody blows on your face or if something's headed towards your face, anything that triggers the stimulation of the eye like that is going to cause you to blink. And so we would call this normally an unconditioned response, and the eye puff would be an unconditioned stimulus. But if repeatedly you play a tone that predicts that this air puff to the eye is coming, then we would have some synapse that must already exist in the brain that will begin to fire at the same time as this air puff neuron would fire, and they both begin to influence this blink neuron. And so if these two begin to fire at the same time, the idea is that this kind of weak synapse, where the auditory system usually doesn't need to activate a blink neuron, becomes strengthened and able to activate that blink neuron in the same way as a kind of a normally strong stimulus like the air puff would be able to do. So when Donald Hebb proposed this, it was around 1950, and we knew nothing. We knew next to nothing about how all kinds of cellular physiology worked, and yet he somehow came up with this really accurate prediction. And the way that he was able to do this was using some really simple logic, but it also made certain aspects of what he predicted, certain aspects of his theory, incredibly testable. So one of the things that he noticed was that conditioning in the brain occurs too rapidly, at least in certain cases, to involve the growth of brand new connections. So it's not as if any time that we need to pair something like an input from the somatosensory system and the auditory system, which are located in different parts of the brain, that we end up sending some part of an axon of a new neuron to grow, which might take a few days in certain cases to traverse the length of the brain and find a path to connect these neurons. It's much, much faster than that. Based off of this, he made some deductions. One thing that he suggested was that this means that these connections must already exist. And as we've seen now, if you look at the anatomy, we have sensory information from our primary areas of cortex that go into these areas that we've talked about over the course of the last 5 or 6 lectures that are kind of polysensory areas. So we know this to be true. Another thing that he predicted is that the connections needed to start out weak and that they must eventually strengthen. Okay, so this suggests that there's some way that we can modify the strength of connections. And then a final couple of predictions are that this change needs to be selective. So whatever the conditioned stimulus that comes in might be, then we need to have something be quite specific. So if I play a tone to predict an eye blink, it shouldn't be the case that I can then play any tone. I need to play back that same 1000 hertz tone, that same frequency or something very, very close to it to get the same response over and over again. And then the final thing about all this selectivity suggests that there must be some sort of very specific convergence between these pathways. And what this means is it goes back a little bit to this idea that these connections must already exist. So we have pathways that are already present and that already converge onto the same neuron or onto some sort of common neuron so that the information from both separate pathways can come together to make a stronger connection. The way that we think that this works is something we actually are going to cover in more depth at the end of all this. But I think as a primer, it makes a lot of sense if I give you an idea of what we think happens. So for many neurons, we have a type of receptor called an NMDA receptor. And this receptor is on many cells, and it's on the postsynaptic side. So it's on the side of the neuron that's receiving information. And these NMDA receptors are notoriously difficult to open because they have a little magnesium ion that blocks them. And the only way that they can become open is if a bunch of calcium is able to enter the cell when something like an action potential happens. And so this means that there are certain synapses in the brain you could think of almost as being silent. or at least silent for the moment. And so what we think happens is that when a weak synapse, one of these ones that's sort of silent and has a lot of NMDA receptors, is activated over and over again, that we end up adding a second type of glutamatergic receptor called an AMPA receptor. And what this does is it makes the synapse stronger, because now when neurotransmitter is released, this AMPA receptor is also a glutamatergic receptor, and now neurotransmitter can combine to this, and it can also bring calcium into this synaptic terminal, and it can activate the NMDA receptor and make it easier for now this dendrite to receive a signal and start an action potential. So what we see is that if we have some kind of learning, that this increases the number of AMPA type glutamate receptors that are at a synapse. And this suggests that the thing that happens with learning is that AMPA receptors are part of the strengthening of a synapse that happens with learning. And as it turns out, if we go back to the fear conditioning example, if we prevent the insertion of AMPA receptors by blocking their ability to come in and make this synapse stronger right here, then we can actually block the response. So if then we play back this tone that is supposed to predict that foot shock is coming, normally animals will freeze, but if you block the introduction of these AMPA receptors, animals won't freeze. They don't form the memory correctly. So we never get this conversion into a long-term memory. So now that we've talked about classical conditioning and some of the ways that we might start to strengthen synapses, we'll also bring in a little bit more here about operant conditioning. Just as a reminder, operant conditioning is either increasing the frequency of a behavior or decreasing the frequency of behavior. So if we have something positive like food, then we can increase the frequency of the behavior. Whereas if we were to give something aversive or negative like a shock, then we might decrease the frequency of a behavior. And the basic idea here is that positive outcomes tend to be associated with increasing a behavior, and negative outcomes tend to be associated with decreasing a behavior. One thing that we know to be really important for operant learning, in terms of neural pathways at least, are a set of connections that we call transcortical pathways. So these are really pathways that are normally important for acquiring, declarative memories that are episodic. And so if you remember from the earlier slides, these declarative episodic memories are the ones where we're kind of telling a story. And so as we're trying to commit something to memory and we're trying to learn it, what we end up doing is something very much like all of you do for all your other memories. You create a story to go along with, or a series of steps. And this could be for any complex task. If you were learning to paint, first you put the paintbrush in your hand, then you dip the paint in the water, and then you dip the paintbrush in the paint, and then you put the paint on the paper, or making a peanut butter and jelly sandwich, or whatever thing that you might be learning that's brand new. What you're going to start doing is by telling a story, and these transcortical pathways are incredibly important for that. Another set of pathways that are really important are ones we talked about a little bit for the motor system, and these are basal ganglia pathways. So remember that these were one of the pathways that was really important for kind of motivated motor behavior. And the reason for this, as we already discussed in the motor system chapter, and a little bit maybe even with somatosensation, is that the basal ganglia is a part of the brain that's getting what we would call efferent copies of sensory and motor information. So what this means is that it's getting information from the primary sensory and motor cortex, and they're converging in one place. So this part of the brain has both of those pieces of information, which means that it knows both what the plan is for movement going forward and then what the sensory feedback coming in is, and it can help to make adjustments. And one of the things that we think is that if you have a repeated sensory and motor pairings, right? So if you're learning to, do a series of steps over and over and over again, then we begin to have kind of common pairings that might happen. And these could come from auditory cues coming in, or they could be somatosensory feedback, or any number of other things that eventually the basal ganglia will get these synapses that are strengthened because there are links now between that tone and that hand movement that you're used to doing, or whatever it may be. And the idea is then the basal ganglia is able to use this information and pass it back into the motor system to help facilitate future motor planning. So not only do we get better at learning this pairing between maybe a cue and an outcome, but we also then are able to strengthen these connections into the motor system so that we can start to get better at performing the behavior. One of the things that we think that the basal ganglia eventually is able to do is to take over and allow certain behaviors, especially stimulus response learning type behaviors, to become automatic. To connect a few of the pieces we've talked about together, the idea here is that we have these transcortical pathways, and we need to be telling the basal ganglia, especially inputs to the caudate and the putamen, what to do. And so we have this transcortical information coming in where we say, first you must do step A, then step B, then step C. And for any of you that have learned a long sequence of steps to build something for the first time, or to complete a dance routine, or any number of other things that require a long series of steps for the first time, then you recognize that eventually you don't have to walk yourself through those steps anymore. This information passes on to the second part of the basal ganglia called the globus pallidus, and really then quickly onward to the premotor and supplementary motor cortex to help change motor behavior, and finally on to the primary motor cortex. What we think, though, happens is that eventually this transcortical pathway isn't needed anymore. We don't have to walk ourselves through those steps, and so you eliminate this, and you just allow the basal ganglia to begin taking over. One of the best examples I can think of this is when you are either walking or driving to work or to class every day, and we have these behaviors where there's a kind of a stimulus response learning component to it. You start out with, okay, I go, left onto Allison Road, and then right here and left here, and all of those pieces end up coming together to be a series of kind of stimulus response actions. But what happens to a lot of people, and I'm certain you've experienced this before, is that at some point we end up going on autopilot. You're supposed to be going to the doctor for that day, and instead you drive all the way to school because you've let your basal ganglia kind of stimulus response system take over. And so the idea there is that this stimulus response learning is helpful in that way. In some ways, we're taking a load off of cortex, and in many cases where being able to do something on autopilot and not thinking about exactly what you need to do when you're driving and being able to hold a conversation is good. But some days this results in you accidentally driving to the wrong place, or walking to the wrong classroom, or whatever it may be. In terms of reinforcement learning, we tend to think of this as being mediated by a number of connections, mainly from dopamine neurons. And when we talked about the motor system, we talked about dopamine neurons in the substantia nigra, but there's actually a second group in a part of the brain called the ventral tegmental area. And the ventral tegmental area is considered to be kind of a reward center of the brain. And it has a few major targets, but one of them is a part of the brain called the nucleus accumbens, which acts as a big input to the basal ganglia. So what we think happens is that dopamine works to facilitate certain responses over others. And this dopamine, we've talked about neurotransmitters a lot, and sometimes I mention this idea of a neuromodulator. So dopamine is considered a neuromodulator, and it's able to help change the activity of other neurons that are firing action potentials. The way that we think that this works is that it fits very much in this Hebbian idea. We might have some sort of a cue that comes in where we might see a lever, and then we might accidentally press the lever, and we get this, these two ideas coming together that would normally create a lever press response. When we add in something like a reward, then we recognize now that there has been something positive, and this helps to come in at this connection where these two would normally come together somewhere in the basal ganglia, like the nucleus accumbens. And what it allows to happen is eventually we can strengthen these connections. And by strengthening the connection, now we can make it more likely that the site of the lever is going to lead to this response in the future. So we start to pair all of these ideas together over time, and dopamine really just comes in to kind of amplify this. The other form of learning that we're going to talk about today is motor learning. Motor learning is yet another kind of learning, and I think this is what most of you would think of as being muscle memory. So this involves either learning to make some kind of a new motor behavior that's very specific, or it involves improving the speed or the accuracy or the fluidity of some sort of motor behavior. So typing on a keyboard or learning to play the piano, things that involve your fingers, which are kind of fine tuned digits of the body, are often really great examples. But it can even be more broad motor behaviors where people get better at a specific sequence that allows them to perform a certain soccer kick, or allows them to engage better the fast twitch muscles for being able to sprint. And this is a mixture of core signals from the brain and from the body. They all have to work together. And much like all the other systems we talk about, what we think of is this involves some sort of amplifying of the synapses for both sensory and motor systems. So we obviously, for many of these systems, they rely on proprioception and sensory feedback. And so by having better sensory feedback, and then a stronger motor output, we can improve the way that we can perform these actions. In terms of the motor cortex structures that are involved, we've talked about pretty much all of these in the motor system lecture, so I'm not gonna spend too much time. I'm just gonna go through these as a reminder pretty quickly. We have the primary motor cortex for controlling movement, the supplementary and premotor cortex, which are both involved in helping to plan these movements, except that the supplementary motor area is involved in planning out sequences, whereas the premotor cortex, as you remember, is involved in being able to guide motor behavior based off of kind of sensory cues or these arbitrary cues in the environment. And then one area that we haven't talked as much about, although we did get into just a brief idea of it, is this ventral premotor cortex. And this is the area where those mirror neurons that we were talking about are involved in kind of imitation learning. So how do we know that this is actually happening? Well, there are a number of earlier studies where they looked at different parts of the cortex, and they looked at the tuning of these areas. And so you remember that if we take something like our fingers, that they're going to map onto a specific space and cortex. And so what the researchers in the study that I'm illustrating here did was that they asked the monkey to work on a task that required them to just use the second, third, and fourth finger. And by doing that, what they were able to do is to actually expand the tuning. So what this means is that the area that used to represent this second digit becomes much bigger. And it actually does this by taking over and encroaching a little bit on the area that used to represent the finger right next door, this first finger. So one of the things we talked about throughout all of the sensory systems is just that the number of neurons participating in some part of the motor system represents the resolution there. So what this shows us is that the brain is actually coming in, and it's taking this single, second digit finger, and it's making it have a higher resolution by kind of giving it more space in the brain. You could think of this in the same way as your television or something like that. If you kind of up the number of pixels, then you make the picture better and more refined. And that's what's happening in the motor system here, is we've given it more neurons to participate, which means that now we can represent this finger a little bit more finely. Moving forward, we're going to get into learning and memory, part 2, and we're going to talk about perceptual and relational learning. So perceptual learning is really very similar to motor learning. It's just the sensory version of it. We're getting better at being able to identify specific sensory inputs because we have practice at identifying them. One really common way that we do this is we get better at identifying voices or faces. And actually, in the lab that I worked at the NIH for quite a few years, there was a long tile floor on the way to the bench where we did a lot of our different chemistry kind of experiments. And a thing that I got better at very, very quickly with all of the different wonderful women in science that worked with me was learning the sound of my boss's high heels compared to everyone else's high heels, so that you knew when she was coming to check in on you, and you could have your progress report and your thought ready to tell her, what the update was for the day. Those little subtle things are all just a form of strengthening those sensory synapses. Part of the reason that we need this perceptual learning is so that we can both learn to recognize new things, and then also we need to be able to identify when something has changed. So this serves us in two different ways. One as a way of forming new memories, but also gives us the ability to better discriminate. So we have sort of an error prediction component to this. And as you all remember, if we're trying to use visual object recognition, then this is going to happen as inputs come into the primary and then subsequently secondary visual cortex. And a lot of perceptual learning for us being visual beings relies on that ventral stream that provides information about what an object is. And our ability to discriminate then relies on some synapse in that ventral stream being strengthened and other synapses not having that same strength. One of the coolest experiments we're going to talk about this entire semester is this experiment right now that we're looking at by Yang and Mansell. And so first I'm going to tell you what they did, and then I'm going to tell you a little bit about why this is so incredibly important. As with many experiments, there's some beauty in the simplicity of this experiment, and yet what it tells us about perceptual learning is incredibly powerful. So what Yang and Mansell did is that they took monkeys and they had them head fixed. If you remember the video we looked at from Hubel and Wiesel, where they had the cats head fixed, and they used a projector to put a picture of a tree onto a certain spot on the retina. And this was in the same location every time, exactly the same location. And so what this means is that only a certain set of cells in the retina were receiving this picture, and that these cells, these photoreceptors and these retinal ganglion cells, as you know, we have this topographic map of what's coming in from the retina. So this means that all the way up through our primary and secondary visual cortex, this information is coming through the same or very similar pathways. And what they found in doing this over and over and over again is that they could make incredibly small changes. So the monkeys learned to differentiate exactly what was happening. And if they came in and added a few yellow leaves or did something like this, the monkeys would recognize these teeny, teeny, tiny differences very, very quickly. However, if they took this same image and they moved it into a different part of the visual field, where now all of a sudden it's coming into a different part of the retina, whether that be left or right or up or down, and then they tried to examine those small changes and see if the monkeys could differentiate those small changes, they were no longer able to do it. So in only this one spot of the retina, where all of the learning had occurred, right here, the monkeys had developed this perceptual strengthening of the synapses. And the reason why this is so important is that it actually proves one of those components of Donald Hebb's hypothesis. So if you remember Donald Hebb, we talked about him last section of this particular lecture series, and what he said was that the synapses that were strengthening had to already be present, but that they also had to be specific. And what this tells us is that because only the cells in this one part of the retina were strengthened, that yes, in fact, those were specific. And that only the specific component of that retinotopic spatial mapping is the one that's having its synapses strengthened and fine-tuned to be able to do that differentiation. And if you go to another spot in the retina, then those cells have not been strengthened to do that same thing. Now, this might seem a little bit crazy, but in the normal world, this is a part of why when we're looking at things, we have a few natural components of the way that we use our visual system that help us to avoid things like this. We're very good about moving our head left to right and up to down. We try and take information in from different angles. And the other thing is, whether you know it or not, your eyes have rapid movements called saccades where they're constantly jittering back and forth in an effort to move the information in your visual field around. So this idea, as I've already said, it supports a very specific component of Hebb's rule. It tests the idea that only a very, very specific set of neurons is being strengthened when we do have this sort of plasticity happening in the brain. And this is probably also why there's been some interesting work that suggests if you go in with an electrode, or if you use transcranial magnetic stimulation in humans and you stimulate the secondary visual cortex, you can actually produce memories. You can cause people to have the idea that they have seen something recently, even though they haven't seen it at all. We can trigger memories because certain synapses are strengthened already, and by activating one area of the cortex, then we can sort of accidentally elicit memories of certain visual stimuli from those strengthened synapses. And this is probably also why if we have damage to certain areas, then we have problems with recognition of certain visual stimuli. And we saw all of these different visual agnosias, or inabilities to recognize things. But we also find problems with memory. And what this suggests, as we've said from kind of the beginning of this learning and memory series, is that memories are not in one place in the brain, but the way that they're actually represented is by this distributed network within the brain, where certain parts of the brain are responsible not only for specific functions, but perhaps also for the memories related to those functions. So one of the things we haven't talked about much is then how we get information from short-term to long-term memory. And there's some evidence for this, although it's still something that we're understanding, that suggests that our prefrontal cortex can provide some input to both the dorsal and ventral stream, and that this helps us to take those short-term memories and put them into long-term memories. And the way that we think that this might be happening is something like that concept of rehearsal we talked about, where if we are really trying to remember something, that our prefrontal cortex may play an important role in helping to maintain attention in a way that is almost like rehearsing the same thing over and over again. So if you are trying to remember that phone number, the prefrontal cortex will help hold that in short-term memory and play the idea back over and over and over again, so that that phone number, if you're saying, okay, I need to remember this, 8675309, 8675309, then you have the ability to internally rehearse these ideas within your brain. The second kind of learning we're going to talk about is relational learning, and this is probably among of all the different components of learning we're going to talk about, relational is almost certainly the most important. The reason for this is that it's the type of learning we think about as being responsible for learning some kind of an arbitrary relationship between two stimuli in the environment. The reason why this is important is because that is most of our world. It's really nice that, sometimes we can develop a motor memory and we can get better at some kind of a task, or that we have a sensory memory. But in that example that I even gave for the sensory memory, there's an arbitrary association. I have to learn to associate the specific kind of click clack of heels with one specific individual, and that's an arbitrary association. There might be some sort of small reinforcing component to it, but in terms of survival and other things, there's really no advantage to remembering that though one pair of high heels on tile is going to benefit me in any major way. It's kind of an arbitrary association. And as you're driving or you're trying to navigate parts of your environment, these are all things that involve that. Spatial learning therefore makes up one of the best examples that all of you can think of. If you're trying to remember directions, you have to remember a completely arbitrary series of stimuli. The example here, left at the McDonald's, right at the church on the corner, and then go to the fourth house on the left. All of those things are eventually getting you to a goal, but they involve along the way this relationship between all kinds of very, very arbitrary stimuli. So as we're going through our world, this is most of what we're trying to remember are these relationships that in many ways have nothing to do with anything else. And this is why I say relational learning is the most important, because it just covers so many different areas of learning that are really hard to categorize specifically into stimulus response learning or motor learning or perceptual. One of the areas that we think of as being incredibly important for relational learning within the brain is the hippocampus. The hippocampus gets input from a number of different areas, including the amygdala, as well as the entorhinal cortex. And these areas are all feeding in information about the value of different stimuli in the environment, but it also has some role in our ability to take in sensory information as well. And this is where we think that these arbitrary associations may be coming from. And on top of all this, we've been able to show for a long time that the hippocampus is important in navigational learning. Some of the best evidence for the role of the hippocampus in spatial learning comes from a task that's been used for a long time in rats and mice called the Morse water maze. And the way that this task works is that a rat or mouse is put into pretty much a giant swimming pool, and somewhere under the water, which is usually colored to be black or white so that the animals can't see through it, there's a platform, and this platform represents safety. And there are two ways that this can be run. In the first case, animals are placed in the exact same part of the maze every time, and they can learn a strategy where all they have to do is go diagonally across the pool, and they can find their platform and get to safety. And the other way that this is run is that you have a variable start position. So the mouse or rat could be put in at 3 or 4 or 5 different locations, which means that they then have to look around the room and say, okay, the safety platform is closest to the door, and so is the door behind me or in front of me, or where might it be? And it turns out that if you give the animals the opportunity to use this constant start position, and you lesion the hippocampus, that animals with the lesion here in blue, as well as the other animals, both perform equally as well as one another. So this means that over time, they learn to get faster at finding the safety platform, and they pretty much perform in the exact same way. However, if you use this variable start position where animals have to learn these arbitrary relationships between things in the environment, I need to learn that the safety platform is closest to the door, and I need to figure out where the door is to figure out then where the safety platform might be, then what you see is a drastic difference in the way that these rats or mice are able to perform, where they take much, much longer to find this same safe platform if you cause damage to the hippocampus. One of the things we also know is that this behavior, this ability to do these relational learning tasks, is dependent on the type of plasticity, this long-term potentiation idea that we talked about before. So remember what we said is that we need NMDA receptors to be unblocked in order to maybe strengthen a synapse. And it turns out if you come in and you block these NMDA receptors, then animals are no longer able to learn the location of the platform, and they swim around for a very long time before they can find it, almost as if you cause damage to the hippocampus. So if we prevent the strengthening of synapses, we actually prevent this relational learning from happening. And yet, if we come in a day later and we inject this AP5 that blocks the NMDA receptors, and we do this on the day after training, then animals are able to swim to the platform with no problem. They perform just the same as before. So what this shows us is that the hippocampus is important for this relational learning, but also that it's transiently important. And at some point, when the animal has had time to learn what's going on, and they've had the time to consolidate that memory to take what might be a short-term memory and then convert it into a long-term memory, that we no longer need the hippocampus. And this is something we're going to continue focusing on. Another really interesting thing that's been found in the hippocampus is through recordings with microwires. So you put microwires into the brain. Remember, we talked about these single or multi-unit recordings during the section on methods. And so if you conduct single unit recordings in a certain part of the hippocampus, it's been found that there are a set of cells that we now call place cells, and that these neurons increase their rate of action potentials for a specific location in the environment. So what you can see in this diagram that I've just drawn the arrow to is that the gray path represents where the rat or mouse has been walking around. They wander around the entire maze. And then in red here, we have the places where action potentials were fired. So each dot here represents a single action potential that's been fired. And so you can see these action potentials only fire in a specific spot in the maze that's pretty well constrained. So we've been doing experiments with place cells for a very, very long time, and we started to learn a little bit more about them. And one of the best studies that has proven that this all has to do with the relational learning was one where they put a rat into a maze that had two sides. So one of these sides was theoretically considered the north side, and the other one was considered the south side. And so if you were to put the rat in this maze, there would be some place cell firing. Let's say that it was only typically on the north side room, that there's a place cell that fires in only one specific spot. It turns out that if you can trick the rat by changing around all of the spatial and relational cues to where you now reverse this compass and you make the rat think that south is north and that north is south, then you can actually switch the firing of that place cell to be in the location that would normally represent the same spot in the other room. And the even cooler thing is that if you then reveal this to the rat and you reverse this back to the other direction, then you actually return that place cell firing back to the original spot, suggesting that once the gig is up, then the neuron recognizes where it should be, almost as if the rat was lost for a minute and then got its bearings and figured out where it was, and then all of a sudden the place cell goes back exactly to the place it had been firing before. Another interesting thing about place cells is that their firing patterns seem to have to do with a little bit more than just location. They seem to have some role in decision making and motivated behavior. So they're more than just simply a location-based cell. They do a whole lot more than that. One of the studies that started to reveal this was actually one where animals were put into a T-maze. So you have a mouse or a rat that starts out in one part of the maze, and what they have to do is run down to the end of an alley and then make a decision to go left to right. And so the food reward that's there is going to alternate, where sometimes it's on the left hand side and other times it's on the right hand side. And so that's their choice. And if you look at the play cells that would normally be firing right here at the end of the maze, then different cells tend to be activated when the mouse or rat is going to make a turn to the left as compared to a turn to the right. And so this suggests that not only is there a relational component here, but there's some sort of a predictive component or a decision-making component that also is part of the input coming into hippocampus to influence these place cells. So the last thing I want to do today is put some of this together and give you an idea of what might be happening with something like memory consolidation. So we have all this sensory and motor information that we know converges into the hippocampus. And the thought is that we process this information so that certain synapses become strengthened through the modification of only specific, very, very specific outputs of the hippocampus. And this allows the hippocampus to kind of play the middleman and act as glue between these inputs and outputs, where only certain circuits, like that Yang and Mansell experiment, become potentiated, whereas others are not. And this means, the hippocampus is now this switchboard making connections between the relational ideas that like to talk to each other and cutting off those that are not necessarily having any conversations. And then, as needed, we can make new connections either through a process called reconsolidation, where we update an old memory, or we can make brand new connections altogether. And this all helps to modify other forms of learning in the brain. This helps us to eventually turn these things into long-term memories, helps to facilitate perhaps some of the sensory and motor type of learning opportunities or stimulus response learning, because we use these arbitrary cues and start to put all this information together.